When you fail a heavy lift, the assumption is simple. Your muscles ran out of fuel. You hit mechanical exhaustion. But if we examine the muscle tissue at the exact moment of failure, it still contains adequate reserves of ATP. The contractile proteins are fully capable of continuing to generate force. The muscle didn't fail. It was turned off. The central nervous system issued a deliberate command to stop recruiting motor units. This intervention is governed by an evolutionary survival mechanism known as the central governor. Its primary function is to prevent any catastrophic physiological damage, like tearing a muscle belly off the bone. The central governor continuously monitors local metabolite accumulation and mechanical tension through sensory nerve fibers. When those thrust markers cross a predetermined threshold, the governor cuts the electrical drive to the working muscle. Your physical limits are dictated by highly conservative biological software, rather than the mechanical capacity of your hardware. To understand how this shutdown occurs, we have to track the inhibitory signal from the brain down into the body. The command for voluntary movement begins in the primary motor cortex as a high-voltage electrical signal, traveling rapidly down the corticospinal tract. But this signal does not reach the muscle cleanly. Along the descent, the spinal cord actively intercepts, filters, and degrades the descending drive. Elite physical performance relies entirely on locating this specific interception point in the spinal cord and forcing the signal through. This animated diagram displays a cross-section of the spinal cord, isolating the ventral horn. This is exactly where the descending nerve signal meets the local motor pool. The final conduit for your movement is the alpha motor neuron. It takes the descending electrical command and fires it directly into the muscle belly to initiate a contraction. Sitting immediately adjacent to that motor neuron is a specialized inhibitory interneuron called a Renshaw cell. As the alpha motor neuron fires its primary signal toward the muscle, it sends a simultaneous impulse through a side branch back to the Renshaw cell. The act of firing triggers its own localized brake. Once activated, the Renshaw cell releases an inhibitory neurotransmitter called glycine, actively suppressing the exact alpha motor neuron that just stimulated it. This chemical suppression dampens the motor neuron's discharge rate. It ensures the muscle fibers never achieve the fully synchronized high-frequency tetanic contraction required to produce absolute maximal force. Your nervous system automatically applies friction to its own output, setting a hard ceiling on your maximum strength. That ceiling is reinforced further down the kinetic chain, at the exact point where the muscle fibers merge into the tendon. This vector model isolates the muscle-tendon junction to show the Golgi tendon organ, or GTO. It functions as a mechanical strain gauge, with sensory afferent nerves woven directly into the collagen fibers. During a heavy, maximal exertion, the active muscle pulls violently on the tendon. The GTO measures this extreme mechanical tension. Before the tension can reach a point of structural failure, the GTO fires an emergency distress signal through the sensory nerves back up to the spinal cord. This distress signal activates a network of inhibitory interneurons, flooding the motor pool with GABA and shutting down the contraction at the source. The body operates on strict biochemical math. It will always prioritize metabolic efficiency and tendon integrity over completing a physical task. These spinal breaks are deeply conserved in human biology, but they can be manually overwritten. You bypass them using a neurological protocol known as concurrent activation potentiation, or CAP. CAP requires the simultaneous intense contraction of remote muscles that have nothing to do with the primary movement. If you are performing a heavy squat, you violently clench your jaw, or crush your grip around the barbell. Here we see a schematic of the descending pathways. That remote intense contraction floods the reticular system in the brainstem, generating a massive secondary wave of neurological noise that descends alongside your primary motor drive. When this dense wave of descending commands hits the spinal cord, it hyperpolarizes the Renshaw cells. Hyperpolarization neutralizes their ability to release glycine. The localized brake is turned off entirely. With the recurrent inhibition disabled, the alpha motor neurons receive an unthrottled, highly synchronous flow of electrical drive, sending a pure signal directly to the prime movers. Concurrent activation potentiation weaponizes your own neurochemistry, manually forcing the body to abandon its safety limits. This reveals the mechanical difference between an average gym goer and an elite strength athlete. The amateur spends their career trying to build physical muscle tissue. The elite practitioner trains the neural signal itself. Deploying cap is not a passive reflex. It requires conscious, aggressive mental focus, perfectly timed to the exact moment of maximal physical stress. Holding that intense mental state under the crush of a heavy load is the only way to force the inhibitory pathways of the spinal cord into submission. The physical result is absolute neural disinhibition. Every available motor unit fires simultaneously, maximizing force output regardless of the muscle's cross-sectional area. Actualized strength is simply the mastery of the mind's ability to overwhelm biological failsafes. The barbell drops not because the muscle tissue physically breaks, but because the mind accepts the central governor's command to quit. Deliberately decoupling the spinal cord through willpower and technique allows you to access a level of force output that borders on structural danger. The absolute limits of human performance are not structural, but neurological, and the nervous system can be broken to your will.